So I am just using the page that was on the front that was with the biomolecules of proteins. And then, oh, it's my awful copying. So the second page, the next one, goes with proteins. But you're going to end up with carbohydrates just like hanging loose again. So we're going to have to make sure we don't lose parts. Okay. Saving the carbohydrates for later. And I'm going to save this little piece of paper. We may use that. Alright. Just heads up, proteins are the longest. So I don't know what order you're going to watch them in, but I know that for me, I'm doing it first because get it out of the way. This is from that second page, and I'm not going to put them side by side. So I'm going to put the first page here by itself, and I'm going to turn the page, and I'm going to delete this big old second page right there. So that second page. I'm almost through with an entire notebook. That's something crazy, because I've had to do, I don't know how many of these videos, like 4 times. So here's the first page. So I don't like how it's laid out. I did it, just saying. This piece right here, what they do, you kind of got to know what they're made out of first. So I'm going to start right down here. I'm not going to start off with what they do. Trying to make myself accountable for doing what I asked you to do. When I decided I started wanting to be neat, probably because I was recording myself. But so I drew my nice little neat line. I wasn't a 12-year-old girl, and I didn't put the things on my eyeballs. So biomolecules. Proteins are a type of biomolecule. As a macromolecule, it has the ability to have a bunch of pieces put together through dehydration synthesis by an enzyme that does the work. And you're going to have a bunch of covalent bonds that are going to be made between the monomers, and you're going to end up with a polymer. That being said, let's really be careful and smart with learning what the actual monomer looks like. And that means it's easy. So every single amino acid has this same basic structure. So it's got this piece, but I'm going to draw it myself. So this is what all amino acids look like. There's 20. So all amino acids have this structure. They have a central carbon. That's what this is. So you start off with that. You have a central carbon. Carbon has 4 bonds. I'm going to go ahead and draw them. There's the 4 bonds. Directly across from each other are going to be 2 functional groups, and you can guess what one of them has to be, because the name of the monomer is based on that functional group. So amino acid, this is the monomer of proteins. So proteins are the polymer, the macromolecule. Their little bitty pieces that they're made out of are all amino acids. So you put 300 amino acids together, and you've got a protein. So you've got every amino acid, the little monomer, has this structure. Central carbon on one side and opposing on the other side, so directly across from each other, because these are spatially locked down bonds. And you've learned that being a tetrahedron, you can't just go twisting this around. It's going to actually have to be in the position it's in. You're going to have what's called a carboxyl side. So that's going to be a carboxyl group, which we learned as COOH. I told you it was a carbonyl and a hydroxyl had a baby. So it's going to be a carbon double bonded to an oxygen, That's carbonyl group with a hydroxyl. So this is the carboxyl group. That's the carboxyl side of the amino acid. Then directly across from that, you have the amine group, which is an NH2. So the carbon's going to be bound to the N, and then you're going to have the two hydrogens off. So this is spatially the same. Now, if I took it and turned it this way, and I have the carboxyl side over here and the amine side over here, it doesn't really matter. The point is that you can't have the amine here and the carboxyl up here. They have to be on opposite sides of that central carbon. And then you have a little lone hydrogen up there. And then you have what we're going to put, they call it a side chain, but that's that R again, which is the variable group. So we're going to come right here and put this part changes. It's the way that you're able to say, oh, I know what amino acid you are, is by this R group. That's what this is called. So it's called an R group. So I'm going to stop this for a second. I'm going to bring up the presentation, and I'm going to walk you through amino acids. I do not expect that you memorize these. I do expect that when you see this, even with the R group there, that you recognize that's an amino acid. You know that amino acids are the smallest piece of a protein. They're the monomer of a protein, and that they have this very standard part here, the amine, the carboxyl, this hydrogen, and that the part that changes is down here, and that's what makes each amino acid unique. So that's what you do have to know. You do not have to memorize these amino acids. But I want to walk you through them, because when you start seeing them, you're going to be able to pattern, and it's going to make sense to you. So we're looking at amino acids because amino acids put together many, many, many in a row, covalently bonded by dehydration synthesis, which is the process, makes a polymer called a protein. Okay, that you do need to know. Okay, so here they are, and they're batched in groups, and we're going to walk through the groups, and we're going to talk about why they are what they are. So, nicely, the majority of them are going to end in INE. So, if you go up through here, and you start counting, there's 5, right? 10, 15, 16, 17, 18, 19, 20. Now, it gets a little weird because people are, I don't know, like there's two forms of these, like you can flip them, and one's bioactives and one's not, and people are, you might want to count it. Anyway, in general, there's 20. A lot like the alphabet. You put them together, and whatever you get by the order you put them together, you can build a protein that's going to do a different job based on the properties of these amino acids. So, let's look through them. This is where we're going to start. There's 4 major categories. There's half of them are nonpolar. That's going to become a really important piece of information. The rest of them, so these do not play nice with water. They are hydrophobic. They do not play nice with water. The rest of these, as far as based on the R group, which is in the shading, the other half either have a full-on charge, positive or negative, or they have a charge that's uneven across the actual molecule. So they have partial positive, partial negative, all built into one molecule like a water. So let's walk through them. I want to start with this one, and I made it ginormous. So we're going to walk through and make sure we understand that everything that's not shaded is the exact same thing. They all have, if you think about the carbon having 4 bonds, 3/4 of the bonds are with things that don't change. So go back to what I said. A central carbon, a carboxyl, even though here it's the ionized form. They're going to show these as far as what they look like when they're in water. But that's still a carboxyl. COO, the H has come off. And an amine group, NH2, but this one's gained a hydrogen. And so it's just the different form of the functional groups that we've talked about. So you're going to always have, with the central carbon, for every amino acid, you have an amine directly across from a carboxyl and just a hydrogen up at the top. What makes this guy aspartic acid is this shaded part right here. So it's going to be called aspartic acid because it has a carboxyl group down here at the very end. Its R group, this variable group, is defined by it's a carboxyl group, and it is naturally going to be an acid. So that's what it does, because it gives a hydrogen away. So this carboxyl group down here defines the way that this whole amino acid behaves. You would think it would be this carboxyl or this amine. It is not, and here's why. When you put these together, so we're going to make a dimer, let's pretend. We're going to put aspartic acid as a monomer together with glutamic acid as a monomer. And we're going to use dehydration synthesis, and we're going to make a covalent bond here with an enzyme. So you're going to lose the ability for this carboxyl and this amine to do anything, because you're going to pull off a hydroxyl, which would be here if it wasn't in solution, and one of the hydrogens would also go, and you'd create a covalent bond here. So these guys are no longer players because they're bound together. The only part that's free in the solution that actually contributes to the properties of the amino acid, therefore the protein, will be the R groups. So this R group is defined by a carboxyl, and so is this one. That's why they have acid at the end. And because once this carboxyl loses a hydrogen to the solution, which is where you get that it acts like an acid, it's going to leave a negative charge. So these are your two negative behaving, because they're charged, amino acids. Okay, let's go to the ones that are positive charge. Again, look right here. Exactly the same up here at the top. Amine group, carboxyl group, central carbon, hydrogen. Now you have 3 amino acids that, because of what they actually have as their R group, they have a positive charge. So methyl, 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 amine group. These all end down here in at least one or more amine group. Because amines act like a base and accept a hydrogen, they're going to gain that proton, which is a positive charge, and these behave like a positively charged molecule. So if I put these two guys together, if I bonded lysine with arginine, I would use the process of dehydration synthesis, and a covalent bond would be made by an enzyme, and we would pull off a hydrogen and a hydroxyl and a hydrogen from here, which would be water. And so the removal of the water would bond these together through an enzyme and make a covalent bond. Okay? So these amine and carboxyl groups do not drive the properties. They are just part of the basic, everybody's got it, everybody's got it. It's like a nose. Everybody's got it. What changes are the R groups. The R groups give the properties to the molecule, functional groups. This R group has a functional group of an amine. So it's going to gain a hydrogen, which means it's going to have a positive charge. Okay, let's go to the polar ones. The polar ones, again, look here, exactly the same. Everybody's the same here above the shaded part. You just plug in the shaded parts, and when you do that, now you've got an amino acid. So methyl group and a hydroxyl. Methyl, methyl, and a hydroxyl. Methyl, hydrocarbon ring, bunch of methyls in a circle, hydroxyl. Methyl, carbonyl, amine. Methyl, methyl, carbonyl, amine. So these have this methyl, methyl, hydrophobic, nonpolar, with these pieces down here, amine, amine, hydroxyl, hydroxyl, hydroxyl. They're polar. So they drive the fact that all 5 of these are like water. They have a difference in charge and unequal sharing of electrons. And so they're going to act like little magnets. So they're not going to just associate with something positive. They're going to have a little bit of the negative part and a little bit of the positive part. And they're going to orient themselves that way so that there's attraction between them. So these 5 are polar. So now we've looked at half. Half are either positive, negative, or polar. All of those go into solution in water. Amazing. No problem whatsoever. The ones that don't play nice are the nonpolar. These do not go into solution in water. In fact, they're going to move to the center of any molecule because of the repulsive forces of the water. Because they do not have a charge, they're not polar, they have nothing. So they don't fit in. It's like when oil floats on top of water. That's an example of the separation of something polar and nonpolar. Oil, fats, are nonpolar. Anything that's carbons and hydrogens, that tends to be where that group, carbon and hydrogen, those are nonpolar things. So let's look here. We can't look at the top. The tops are exactly the same. Your R groups, the easiest, smallest, tiniest amino acid there is glycine. Its R group is just a hydrogen. It is the smallest amino acid. But hydrogen and a carbon, or hydrogen by itself, nonpolar. So nonpolar, methyl group, nonpolar. 3 methyl groups, nonpolar. Methyl group with a sulfhydryl. Sulfhydryls are still nonpolar. Methyl, methyl, methyl. Methyl, 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 methyl. Like I'm saying names. Methyl, 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 methyl. Methyl with a sulfur. Now that's not a sulfhydryl. It's just a sulfur. And then a big ring of carbons, hydrogens, and a nitrogen. So no oxygens at all. That's the big deal for nonpolar. No oxygens, just carbons and hydrogens. Maybe a little sulfur here and there, and maybe a nitrogen here or there. And then here, methyl group in a big, huge carbon hydrogen ring. So the takeaway for all of this is, nobody's asking you to memorize these, but it's something you should be able to do is, first of all, you should be able to look at this molecule if it was given to you and go, I know you're an amino acid. I know that because amino acids all have a central carbon, and 3/4 of their bonds are set and do not change. Amine group, carboxyl group, and a hydrogen. This variable 4th bond is the R group, and it tells me about the amino acid. Methyl, methyl, methyl tells me it's nonpolar. You won't have to know the name. You wouldn't have to know any of that. But you should be able, we have little cards, little flashcards, and you should be able to look at it and go, amino acid. I know what this is. This is a monomer of proteins. Phew, that was a lot, and we didn't write anything down. So now we should be much more comfortable with this structure that we drew. And I told you, you wouldn't want to be called, hey student, get over here. So every one of these polymers, so we're going to come over here and write, be very specific about what the polymer is called. The monomer is called the amino acid. The polymer is called a protein. The actual covalent bonds, we're going to give them their own name, and they're called peptide bonds. So peptide bonds are the covalent bonds between each amino acid. Amino acid's the monomer. So the covalent bond is made by dehydration synthesis, and it's all of them have it. All of them have covalent bonds. But we actually name them based on what polymer they're part of. So yes, it's a covalent bond, but we go ahead and we give it its own name called a peptide bond. That being the case, so let's come over here now and do some more just writing over here. When we're talking about proteins, so that's where we're working. The polymer, they'll either call it a protein or they'll call it a polypeptide, named after the peptide bond. So, or a polypeptide. They're the exact same thing. Nothing different. The monomer, each individual piece, is an amino acid. And there's 20. And the order matters. We're going to talk about that. So, you've got proteins. They're made up of amino acids. The name of the covalent bond, so the covalent bond that's between amino acids is called a peptide bond. So you would have amino acids that are covalently bonded together through dehydration synthesis. This is called a peptide bond. And this whole thing, the polymer, is either called a protein or a polypeptide. Now, obviously, that one's short. Okay? Oh, this is a huge, big piece of information. I'm still not going to do this top piece yet. Turn the head. Okay. I've said this a billion times. Function depends on structure. Change in structure, change in function. And you get the function based on the structure. So, I'm going to use my little piece of paper. And I'm going to cut it. And I'm going to make a polypeptide with you. And I think what I'm going to do is I'm going to go, I know, crazy person, I'm going to go back to the amino acids, and we're going to use very specific ones, and we're going to put them in order, and I'm going to show you what happens based on each amino acid's property. Okay? Let me go open that up. So let's make a polypeptide. Let's pick some amino acids and put them in order. Now, I'm going to write mine on my paper. You can write yours on your paper once we come back to being able to see in mine, because I will have the document camera up. So I think what I'm going to do is I'm going to have a couple of non-polar to begin with. So all I'm going to do is I'm going to write, this is what you write. Now, the actual letter is fine, but most often people are going to use this three-letter name. So I'm going to start off with the tiny one, with glycine, And I'm going to remind myself that it's nonpolar because I'm not going to draw it out. And then let's have a valine. And then let's do some other things. So let's see here. I'm going to, sure, why not? Let's have histidine. Let's do histidine. And it's got a positive charge. Let's see. What? Ooh, let's do some polar ones in the middle. Let's do serine. Let's do tyrosine. And how about getting a negative one? Okay, I'm obviously not driving this correctly. There. So we'll do aspartic acid. And then let's see what I want to do. Let's put another couple of polar ones. So we can do threonine. Actually helps you sleep. It's polar. Let's do, oh, I don't know, arginine. It has a positive charge. And then I want to do some more non-polar. So let's do leucine. And let's do isoleucine. I can't even see. Oh, I know that's not what that is. It's okay, non-polar. Okay, so I'll come back to my little part. So this is what I drew. So I put some amino acids in order. So I'm going to just have them make sense to me. I'm going to make them their own individual thing, because these are my monomers. And I'm going to, because I have been doing this, covalent bond them together. So now I have a dimer, trimer, and I'm beginning to make a polymer. There we go. So those are my covalent bonds. They're called peptide bonds, because that's what covalent bonds in a protein are called. And I'm going to remind myself, ooh, let's use blue, that when these were made, each one of them, you had dehydration synthesis, which means a water was removed. So I'm just making myself just a step-by-step way of remembering. So there was an enzyme that put those two together through dehydration synthesis, and the water was removed and they became covalently bonded to each other, and I'm calling that a peptide bond. Now, this right here, this order is called a primary structure. So let's write that in there. So the primary structure is the linear order of the amino acids. It's the sequence that you have. If you sequence the protein, it's where it would come out with the order of the amino acids, what was bonded to what. And order matters. Okay? So right through there, that is my primary structure. Now, it does matter what goes with what, because this molecule is going to interact with itself, and we're going to get folding. So right through here, now we're going to have some folding. The secondary structures, those have to do with making one of two shapes. So secondary structure is either you are either going to make one, you're going to make an alpha, which that looks like this, helix. So if you've ever seen a ribbon model of a protein and you see something that looks like this. See that? Like a little ringlet? Like a ribbon? It's going to have a coil. Look, that's an alpha helix. It's a region, or what we call a domain, that has that structure to it. So now as the protein is no longer linear, parts of it will begin to coil, and it's going to make an alpha helix. The other type of common secondary structure is called a beta, and this time it's written like that, sheet. And it's going to be like a pleat. If you've ever seen a pleat in a clothes where they bring together and they pinch together, that's a pleat. Okay, that's what it's like. Sometimes they'll call them a pleated sheet. So these are going to be based on, all of these are associations with amino acids that are close together. So not way apart in that linear order, but that are close together to each other. They associate with each other, and the reason why they make these shapes is because of hydrogen bonding. So you get some of that polar with polar, and it'll begin to make some hydrogen bonds, and it starts to make some of these just runs of shapes. So primary structure, tell me the order of the amino acids. Okay, well I put glycine, valine, and then there's histidine, and just what are they in order? Each one though has a functional group, and it brings with it the behavior of that functional group. So because of that, with these polar pieces, those are going to begin to have associations, and so you have a secondary structure. It is based on the linear structure because the hydrogen bonds that hold this together, the alpha helix and the beta sheet, are because of the properties of the amino acids. So then you begin, the tertiary, you begin to get a 3D structure. So tertiary structure So right now, this is technically 3D, but you're not really doing anything that's unusual. All proteins have alpha helixes. All proteins have beta sheets. But here is where you get the unique 3D structure for that protein. And this is based on R groups. So this is from tertiary structure is because of the R groups of the amino acids. You can't really see that. What have I got that's better looking than that? Nope. I have so many pens and I don't know why I've got a problem. Let's do blue. Is that better? Yes. So it's from your R groups. Remembering your R groups are either positive, negative, polar, or non-polar. And your amino acids, it's like finding each other, long-lost friends. So let's see what we've got here. Let's read this. The polar, the charged, are going to very nicely associate with water. The non-polar, so all of this, everything but the non-polar, will face the water. The nonpolar, so let's just put a little note, key. Nonpolar is drawn to other nonpolar. Nonpolar means hydrophobic. So it's going to move away from water, and they will together dive to the middle, because now we have a middle, because it's 3D. Drive. What? Dive. To the middle of the protein. So, nonpolar on this end and on this end, they're going to find each other. Okay, so they're now associated with each other. I've got the nonpolars with the nonpolars, and they're going to dive to the middle of the molecule. Everything that's polar or charged will stay on the outside. So this is going to dive into the middle, and now you've got a shape. So these nonpolars are moving into the middle, moving away from the association with water and leaving the polar and the charged to be exposed. So now you've got a 3D shape, and it's based on the R groups, which the R groups, when you say that, it's going to be based on the functional groups that they have, which make them either positive, negative charged, polar, or nonpolar. Nonpolar dive to the middle. And I don't know why I put this one up here with it. Now I'm going to have to rewrite it. Okay. So, quaternary just means that you have a polypeptide, which is what this is now, and you have more than one. So quaternary just means you associate more than one polypeptide together. I guess I could have made this screen full size. I'm a terrible videoer. So this just means that you have more than one polypeptide together. Hemoglobin has 4. So it's like, how many subunits do you have? You get to the point now where you have subunits. You have a couple of polypeptides together. This is a long video. Okay, so you have to understand and know the different levels. That's what this is called. So this is levels of structure. Proteins have levels of structure. Honestly, it's all driven by the linear, by the primary. The order you put them in determines later which ones go, oh, you're real close by. I'm polar, are you polar? Yeah, we're both polar. Let's make some alpha sheets. Okay, how about, or alpha helices, what about a beta sheet? And then your R groups then cause the intricate across the protein, big distances apart folding. And your nonpolar dive to the middle, and the rest stay exposed. And then the quaternary just means you've got more than one of them together. So all of this levels of structure, that's going to give you the unique function. Each one that has a different linear order will have a different shape. Shape is essential. Speaking of that, what happens when you lose your shape? You don't function. So, denaturation is going to be the loss of the structure of a protein. It unfolds. We used to call it melting. but that was a bad way of putting it. So it's the loss of the structure, which equals loss of function, and it just unfolds. You've got some situation that it's in, and you're overpowering these other interactions. Something stronger than hydrogen bonding or the association of the R groups is going on, and the protein just can't stay stuck together anymore. So it's going to go from looking something like, okay, so let's have some alpha helices and a beta sheet, and then another run, and then some more alpha helices and some beta sheets, and then you're going to come in here and come over there and make some loops. You're going to go from this to that. So when you have denaturation, you lose everything except the primary structure. Because they're still in order. You still have your amino acids in order. They haven't all fallen apart. Those covalent bonds are tough. I told you. You have to purposefully break them. And this is a lot of times based on pH and heat. So you lose the quaternary, the tertiary, the secondary structure. But the primary, the order that they're in, remains. Now, most of the time, if you put this unfolded protein back into a good environment, it just folds right back up. So this is reversible, the folding and the unfolding. This is all based on this denaturation is going to be usually caused by temperature range that's not good for the protein. So a change in temperature, and you get out of the range where the protein can handle the kinetic energy, which is what temperature is, and or a change in pH. Because pH is hydrogen ions, and if you get down to a really low pH, those hydrogen ions are positive charges. They disrupt all of this because there's just tons of charge in the water. Or if you have a high pH, there's tons of hydroxyl, there's tons of charge in the water, and it just can't hold its shape in all of that. Okay? Believe it or not, we're almost done. This is an important picture. This demonstrates for you how that this enzyme can break a covalent bond, and you end up with the monomers being separated. So sucrose is a dimer of glucose and fructose, so this is table sugar, and there's an enzyme, sucrase, named because sucrose sits in there. Nothing else can, just that. And when this enzyme sits there, this is because you're going to add water, because this is called hydrolysis. This is the process where we break the bond, by adding water back. Hey, you want your hydrogen back? You want your hydroxyl back? Good. Now go your own ways. So you give back the water in the form of a hydrogen and a hydroxyl across that amine and carboxyl group right here. And when you do that, they're separate again. So this is the process of breaking the covalent bond. And what did we call that covalent bond? For proteins, also known as a peptide bond. So, we'll do enzymes, I think, by itself. So we're going to put here C, enzyme notes. Okay? Only thing we have left is, okay, I got it. here, I have a polypeptide, and it's in this shape because of the order of the amino acids, the association close by, secondary structure, alpha helix, beta sheet, hydrogen bonding, associations way across it where the R groups come together, and you get the 3D shape where the hydrophobic nonpolar dive to the middle, everybody else stays on the outside, and here's my folded protein, and it has a structure, therefore a function. What function? Keep telling me, structure and function, structure and function. What do proteins do? Glad you asked. This is what they do. So they have many functions, but primarily enzymes are proteins. So this is just a special type of protein, and its job is to catalyze, which just means make it happen right now, reactions. So it's going to catalyze reactions. So what we want to be careful about is we don't want to, they're so important, enzymes are the big deal. So if I have two molecules, and an enzyme's job is to put these together. Here's the deal. If you give this enough time, and these molecules just move and orient themselves in space, eventually, at some point, statistically, small percentage, but they'll orient themselves like this. Enzymes are like a matchmaker. Here, come here. You come here. You come here. And they put them in a proximity, so what would naturally happen happens quicker. So enzymes are going to catalyze reactions. So just think they're like a matchmaker. Have y'all watched that new series on Netflix? I'm going to have to watch it. It looked really cool. So think matchmaker. Or if you have an enzyme that's job is to break these apart, these bonds, like I told you, covalent bonds, you have to purposefully break them. The enzyme holds the molecule and puts torsion or torque on this bond, and the bond can't hold based on the pull against it, and it breaks, and then you have this. So this is synthesis, right? Anabolism, putting together, building. This is lysis, breaking, catabolism. Enzymes do that. Enzymes are going to do that. Proteins are structural, so they're just going to give shape. And they're going to, like if you think about an example would be like cellulose in the cell wall of a plant. So it's going to give structure, shape to something. It's going to just be structural. It's not doing really anything but holding the shape, which that's super important because I don't want to be a big blob of goo. I'd like to have some shape. So that's the second thing that proteins do. The third is transport. So often we have molecules that are too big or have a charge or cannot go by themselves into or out of the cell. And you have a protein that's like a gate. So it's going to let molecules transport or move across a membrane. So transport would be, you could have a transport protein is like a door or a gate in the cell membrane. And so things that are too large or charged and wouldn't normally come across, or it could be molecules like kinesin, which actually is a super cool molecule. It's like got a foot, and it actually transports things by walking. So those are examples of proteins that have the job of movement, transport. Proteins also have the job of regulation. So sometimes what they do is they get in the middle of processes, and they either are necessary and you have to have them and things won't work without them, or they're in the way. So they regulate, so they have a role in a process that's required. So if you want to actually take a gene and get the product from it, you have to have transcription factors, which are types of proteins that have to show up and help the gene be transcribed. So an example, transcription factors. Defense, we get into this later in the spring, but we're all hearing about it right now. Defense would be part of the immune system. And I'm going to go give blood at Marsh, and if you do, they do a serum antibody test to see if you've had COVID. Because sometimes you don't know you've had it. And if you've had it, you have antibodies to COVID, which not COVID, it's the coronavirus. COVID's actually the disease. But you would have antibodies, which are a type of protein that's part of the immune system. They're made specifically against one pathogen. Storage. Proteins have a job in storage because sometimes they're going to be like a reserve or a place where that other molecules are kept. So an example would be ferritin, and it's a protein that holds on to iron. So it's protein that holds iron. So storage would be where that it would be about reserves or holding on to something. And as usual, there's one that I forgot. So we're going to put a little note up here and put 7. And it's signaling. So signaling would be for communication. And a lot of molecules serve as a signal, and they sit in a receptor, and they're going to cause a change in behavior in that cell. So an example would be a hormone. Hormones are proteins, and they go and they communicate to the cell to go do something. Hormones travel through the blood, and they go like a lot of times from the pituitary, and they travel to all the cells, and then they tell the cells through communication by sitting in the receptor to have an action. So this is going to be a stimulus with a response, and the signal is the stimulus. They're not all this bad. Proteins are the big part of macromolecules. I did warn you when I said that this was nothing like what you did in biology. So I'm going to put a few little videos after here so you can hear. Again, I strongly suggest that you use a different color pen and you add on, because proteins never go away. They're big, big, big deal. All right. I think you're wonderful.
Hey, this is Mr. W. Welcome to Thinking Biologically. This video is about amino acids and protein structure. The goal of this video is for you to be able to explain the structure of amino acids, peptide bonds, and the 4 levels of protein structure. It's going to be much easier to understand all of this if you're familiar with functional groups. If you need to, head on over to my functional groups tutorial and video at sciencemusicvideos.com. The monomers of proteins are amino acids. Each amino acid has a central carbon, sometimes called an alpha carbon. This central carbon is attached to an amino functional group, a hydrogen atom, a carboxylic acid functional group, and something else. That something else is represented by the letter R, and it's called an R group or a side chain. Each of the 20 amino acids has a different R group, and based on the R group's chemistry, we can organize amino acids into one of 4 types. Some amino acids have non-polar or hydrophobic side chains. Just look for a methyl group or a carbon ring. Some are polar or hydrophilic. You can tell by the presence of hydroxyl or sulfhydryl or carbonyl. If the side chain ends in a carboxylic acid, it's acidic and it'll have a negative charge. If it ends in an amine, NH3, NH2, or NH, all with a plus sign, then it'll be basic and positively charged. Here's why this is important. In cells, DNA makes a molecule called RNA, which tells ribosomes how to make proteins. Mutations in DNA can wind up substituting one amino acid for another. If the change results in the same type of amino acid, non-polar for non-polar, then that mutation might not be a big deal. But if you change the type of amino acid, that can be a very big deal. The inherited genetic disease, sickle cell anemia, a blood disorder characterized by tissue damage and pain crises, is caused by a single mutation that substitutes valine, which has a non-polar side chain, for glutamic acid, which has a negative side chain. Proteins are polymers of amino acids. The bond between the amino acids is a covalent bond, and it has a special name, a peptide bond. This animation shows in a cartoony way how these peptide bonds are formed by ribosomes during protein synthesis, a process we'll learn much more about in another video. The reaction combining amino acids is a dehydration synthesis. Focus on number 2 in this diagram. The ribosome removes this OH on the carboxyl group of one amino acid and the H on the amine in the second amino acid. The result is a peptide bond, and it's shown at number 4. Number 6 shows the resulting water molecule. With more amino acids, you have a polypeptide. Note the internal sequence of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That can go on for hundreds of repeats, and that's the polypeptide backbone. Is this making sense? Please leave me a comment down below. Maybe something like, amino acids are awesome, or ask me a question. Then please go over to Science Music Videos and do this quiz. It's in the card above and it's linked below. It'll solidify your learning, then come on back because the really cool stuff about proteins is coming up. What you're looking at is a protein that's called an antibody. It's part of our immune response. Focus on the shape that's at the tip of the arms. That shape enables the antibody to bind with surface molecules on the disease-causing agent, like a virus. Once the virus is coated with antibodies, it's neutralized and it can't attack any more cells. The point is that antibodies can only do this if they fit those surface molecules on the virus as specifically as a key fits into a lock. The shape is everything. Now let's think about muscle contraction. There's specificity and there's also a capacity for the shape of the protein to change. Muscles contract because of the interaction of 2 proteins, actin and myosin. Myosin is like a powered molecular hook. It can reach out, grab onto actin and pull. That causes the muscle fiber to shorten and contract. Protein structure comes about through 3 or 4 levels of molecular interactions. These are referred to as levels of structure. The first level is primary structure. It's the genetically determined sequence of amino acids in a polypeptide chain. In this diagram, the sequence is leucine, lysine, glutamic acid, serine. That sequence was encoded in DNA, and it can involve hundreds or thousands of amino acids. Secondary structure comes about from hydrogen bonding between the carbonyl groups and amine groups within the polypeptide backbone. The carbonyls in this diagram are at 3 and the amines are at 2. Remember that these functional groups are polar and they have opposite charges. One of the shapes that can emerge from this kind of bonding is an alpha helix. It's a corkscrew, and it's held together in this shape by hydrogen bonds between the carbonyls and aminos that are 4 amino acids apart in the chain. The other form is called a beta-pleated sheet. A pleat is this. It's a series of regular folds. This happens when parts of a polypeptide chain run parallel to one another, like this, or anti-parallel, like this. And that allows the carbonyl and amino groups that are in the different parts of the chain to interact, forming this kind of pleat. Now, How could the polypeptide chain fold back on itself or become parallel? Because of tertiary or third level interactions. Tertiary structure, third level structure, comes about through interactions between R groups. This causes the polypeptide chain to twist and bend, and that can bring sections of the polypeptide that are far away from one another in terms of sequence spatially close to one another, so that you can have structures like beta-pleated sheets. In this diagram, number 1 represents the polypeptide backbone. NCC, NCC, NCC. At number 2, a hydrogen bond has formed between two polar side chains that end in functional groups with opposite charges. That hydrogen bond is causing a bend in the polypeptide chain. At 3, we have a disulfide bridge. This forms whenever cysteines, which end in sulfhydryl groups, get close to one another. Their sulfhydryl groups form a covalent bond between the two sulfur atoms. This causes the polypeptide chain to loop back upon itself. At 4, we have hydrophobic clustering. You can think of these as being like oil droplets within the structure of a protein. These hydrophobic R groups are avoiding water, and they're also forming very weak bonds with one another. At 5, we see ionic bonds forming, again causing a loop. Combine these tertiary interactions with secondary structure and very complex shapes can emerge. Here's myoglobin. It's an oxygen-carrying protein within muscle tissue. This shape was set down by the primary structure, which is genetically determined, which led to these alpha helices, a secondary structure, and these bends and turns caused by tertiary interactions. For many proteins, that's it. But for some proteins, there's yet another level. This is the quaternary or fourth level structure. It involves multiple folded polypeptide chains bonding with one another. You can see this in hemoglobin. It consists of 4 polypeptide chains, and they're connected to one another through any of the bonds that occur at the tertiary level. What we've covered explains the specificity of protein shapes. But what about the ability of proteins to change their shape? Here's a cartoon of a membrane channel with an attached receptor. The channel is a protein, and it's closed until a hormone binds with the receptor. Then the channel opens up, allowing some particular substance to diffuse through. How does this work? The channel is built out of alpha helices that wind their way through the membrane. Alpha helices are molecular springs. When the hormone binds to the receptor, it's like pushing on one part of a spring. This change ripples through the entire structure, in this case, causing the channel to switch from closed to open. The big idea? Proteins are molecular machines. Here's one called ATP synthase. Powered by flowing protons, it has internal parts that spin around like a rotary engine, catalyzing the formation of ATP, life's energy molecule. Here's one that synthesizes DNA. Last point. Because much of protein shape is determined by interactions like hydrogen bonds and ionic bonds, the shape of a protein can easily be disrupted. Change the pH a protein is optimized for, and its shape deforms. Why? Because the new pH might disrupt ionic bonds at the tertiary level. Too many ions can do the same thing. Too much heat can disrupt hydrogen bonds and cause other changes. That's what happens to egg white when you fry an egg. These kinds of disruptions that change a protein shape are called denaturation, and they can cause proteins to lose their function. Sometimes denaturation is reversible, and sometimes it's not. At my website, there's a quiz that will help you master all of this material. Please go there now. Remember, the first 2 days on my site are free, so it doesn't cost you a thing to try it out, and it's the best investment you can make in your biology learning. Here's some important things.
Hi, this is Ms. Delosier, and these are your notes on proteins. I'm going to try and keep these short, but I'm not making any promises, y'all. So proteins are the most structurally and functionally diverse group of macromolecules. And so they have a bunch of different functions, and we need to talk about a bunch of them. So the first thing you need to know is that all enzymes are made of proteins. So enzymes are biological catalysts. So They do things like they make DNA, they break down lactose, they are responsible for making your ATP. So anything that ends in this suffix ase is an enzyme, and that means it's basically doing something in your body. It's catalyzing some reaction. They're also structural, so like collagen and keratin are structural proteins. They are used for transportation. So hemoglobin is a protein in your red blood cells that moves oxygen, and you have protein channels in your cell membranes that move things in and out of your cells. They are used in cell communication pathways, so they're used as signal molecules. Hormones like insulin are proteins. Some receptors, not all receptors, but some receptors are proteins, and they're antibodies. So we've been hearing a lot about, antibodies in the news lately. Well, antibodies are specific proteins that your body makes to go ahead and defend. It's part of the immune response. And I'm not going to go into it because these notes are long enough right now. You just need to know that antibodies are part of your immune response. And then movement. So some proteins are involved in movement, specifically actin and myosin. So let's talk about protein structure. So proteins are polymers that are a specific type of polymer. They're called polypeptides, and they are made up of monomers, like all polymers are. And the monomer of a polypeptide is an amino acid. Okay, so an amino acid has, there's 20 different amino acids, and they have an amine group, they have a carboxyl group, and they have what's called a functional group. So this N bound to the H2, this NH2 right there, that is your amine group, and the COOH, that is your carboxyl group. You don't really need to know that. You just need to recognize that that's an amino acid. And then they are also going to have a functional group. And the functional group is the only thing that's different between the 20 amino acids, and that just hangs off that center carbon. And so like R is just generic. Like it could be like a giant thing, but it's just whatever's different between the amino acids that hangs off the R group. So how do I form a polypeptide? Well, of course, you form it through dehydration synthesis, like you form all polymers. So I'm going to take one amino acid, and that one, it happens to be glycine. And I'm going to take another one, and that one happens to be alanine. And I want you guys to notice that the glycine and the alanine have different R groups. Like one is just a hydrogen and the other is a methyl group, right? That's what I mean by functional group. It's just what's hanging off in that circle at the top, right? So I'm going to remove the water, and then that's going to give me a dipeptide, right? Two peptides covalently linked together. But in proteins, we don't just call it a covalent bond. No, because proteins have to be difficult. We call it a peptide linkage, but it's just a covalent bond. It really is. But that's a peptide linkage. So if you hear something that says, oh, well, this face cream contains peptides, that means it contains little short chains of amino acids. So I mean, I don't know if we really need to pay that much more for expensive face cream because it has peptides in it. I don't know. So let's talk about protein structure and function, because the reason that proteins can have so many different functions is because the structure of proteins varies so drastically. And the reason is that the structure of a protein dictates the function. The structure dictates the function. The structure dictates the function. Like I can't stress how important that is. So the 3D structure is unique, and if it's altered, then that changes the function. So depending on whatever the overall 3D shape of your protein is, that's going to determine what it does, right? So we have to talk about how that works. It's basically like a lock and a key. Like if you took a key and you bent your key or you filed part of your key off, it's not going to open the same lock. So there's 4 different levels of protein structure, and I'm going to go over them super fast. Do not take notes on these. I need you to hear me say it. I want you to try and understand it, but this is like biochemistry, AP biology. This is related to anatomy and physiology because it determines level of structure. It is technically part of your course, but in no way, shape, or form am I going to ask you about these on a test. All right, so there's 4 levels of structure and function. There's the primary structure. The primary structure is just the amino acid sequence, just the order of amino acids that you got. So like methionine, alanine, serine, lysine. That's it. That's all it is. It's determined by the actual DNA that's transcribed and made into a protein. That's transcribed in translation. It's just determined by the DNA transcription translation. Okay, so that's it. It's linear. It's covalent bonds held together by peptide linkages. The secondary structure is caused by local folding. So you have short sections of the protein that fold and form hydrogen bonds between the amino and the carboxyl groups. So this doesn't involve the R group at all, right? What does that mean? I don't know what you're talking about, Ms. Delosier. Basically, little sections form like this little spiral alpha helix or this little pleated beta sheet. That's all it means. So you're going to get little short sections of your overall protein that fold up and make spirals and like little fan shapes. And so that's your secondary structure. It does form like a 3D structure, but like not the complete 3D structure. It just starts to, instead of being a straight line, it starts to spiral up and kink. The next one is your tertiary structure. And your tertiary structure is like how the whole molecule folds up. And so like you end up getting like interactions between hydrophobic parts of the molecule and hydrophilic parts of the molecule. And so the hydrophobic parts want to be together and the hydrophilic parts want to be on the outside. And so it basically just folds up and you get a 3D molecule. Sometimes you get like some sulfur molecules or some charged R groups that are going to go ahead and like form secondary bonds. No one cares about that right now, but basically it's just the 3D shape. So it kind of forms that 3D structure, the globular shape, the overall globular shape. Globular is actually a real word because like when we start talking about immunoglobulins, they're talking about the globular shape. Okay, and then quaternary structure, that's the 4th level, is when you have like multiple polypeptides like hanging out together. So like for example, hemoglobin is actually made up of 4 protein subunits. It has 2 alpha subunits and 2 beta subunits, and all 4 of those subunits go hang out together to make the hemoglobin protein, which carries oxygen in your red blood cells. So, and it's based on hydrophobic interaction. So basically just the proteins want to go ahead and like if both of my palms are hydrophobic and they don't like water and I'm floating around in the cell, look, now I don't have to be around water. So that's all that means, hydrophobic interactions. So the quaternary structure is actually what determines the overall function of the protein. The final 3-dimensional structure determines the function. And so that's the super 3D structure. So I know that was a lot. So hopefully you understood that. The big takeaways on proteins are actually what they're used for and that they're made of amino acids. The rest of it, I just kind of hope you start thinking about when we talk about other things in class. Obviously, as always, if you have any questions or need any help on clarifications or anything, just go ahead and reach out to me and we'll set up a tutoring time. Thanks. Hi, this is Ms. Delosier, and these are your notes on protein folding and denaturation. So the first thing we need to talk about is protein structure and how that relates to protein function, because that's going to be mainly why we talk about protein folding. So this is super important. The structure of a protein dictates the function of a protein. The structure dictates the function. Structure dictates function. So what does that mean? Here's what that means. If I have a protein that's job is to bind to starch and to break down the bonds between the individual glucose molecules in the starch molecule, that protein is shaped so it fits into that space between those alpha glucose molecules, and it can go ahead and catalyze, speed up that reaction to go ahead and break down that bond. And if I have a different shaped protein, it's not going to fit in that space. And that's one of the reasons that we can't actually break down cellulose, because that amylase enzyme doesn't fit into the shape where we've got those beta glucose molecules. So the structure of a protein dictates the function, and you're going to find the structure dictates function throughout biology all year long. So the 3D structure is unique. And if I change that structure, if I alter the structure, that's going to alter its functionality. So if I take and I change the shape of a hemoglobin molecule, that's going to make it unable to carry oxygen as well as the normal hemoglobin molecule. So you can kind of think of this like a lock and a key. If I have my key and that's my protein and I change the shape of my key, is it going to open the lock? No, because it's specific to that lock, right? So there are 4 levels of structure that we talk about in proteins, and we're going to talk about each of these individually. The first level is the primary level, and the first level of protein structure is really easy. We've talked about it a bunch already. It's just the sequence of amino acids. So for example, my sequence could be methionine, alanine, serine, lysine, and that's it. I mean, it would be a lot longer because proteins are a lot bigger than 4 amino acids. But there's my example sequence, and that is determined by the DNA. So your DNA is transcribed and then translated, and that determines the order of the amino acids in a protein. And those amino acids are going to line up in a linear strand, and remember, they're held together by covalent bonds. That peptide bond between the amino group of one amino acid and the carboxyl group of the other amino acid, those are actually going to be covalent bonds. So that's my primary structure. My secondary structure is going to be local folding. So I'm looking at really short sections of my protein, and you're going to get hydrogen bonds that happen between the backbone atoms. So really you're going to get hydrogen bonding between like the amino groups and the carboxyl groups, because if you look, the molecule kind of becomes polar, and then as it folds up, you're going to get some sections that are going to be positively charged and some sections that are negatively charged. So it's all going to form a 3D structure, and it's primarily going to form this corkscrew shape, which we call an alpha helix, and then it's going to form this shape, which is called a beta sheet. So think of it when you take a piece of paper and you fold it up to make a fan. That's a beta sheet. It's actually a beta pleated sheet, but sheet is fine. So the 3D structure, and that's your alpha helices and your beta pleated sheets. Localized 3D structure, alpha helices, beta pleated sheets. So if I ask you for an example of secondary structure, alpha helix, beta pleated sheets. That's it. And that's all due to those hydrogen bonding. The tertiary structure is where I get my whole molecule, my whole protein structure. And it's going to be due to hydrophobic and philic interactions. But these aren't going to be hydrophobic and hydrophilic interactions between the central atoms of the amino acid. They're going to be between the R groups. So those R groups, remember I have polar and nonpolar R groups, and then I've got positively and negatively charged R groups. So I'm going to get hydrogen bonds forming. I'm going to get some ionic interactions between positively and negatively charged R groups. And then when you have those amino acids that actually have an R group that contains sulfur, you can actually get what's called a disulfide bridge, where the sulfur atoms in your R groups actually will form bonds between them, and those kind of lock and anchor the 3D shape together. Really, all the tertiary shape is locked and held together by these different types of bonds. So it's my overall 3D structure. Tertiary, third level, is the overall 3D structure, and it's the overall globular shape. So when I look at just a picture of the protein, what's that overall 3D shape? That's my tertiary. My quaternary structure is my fourth level, and it happens when I have multiple polypeptides that are going to go ahead and interact together through hydrophobic interactions. So those multiple polypeptides are going to kind of group together to keep the hydrophobic parts on the inside so it's not exposed to the surrounding fluid that is high in water. And remember, that's a polar molecule. So I want to go ahead and have those nonpolar parts kind of touching of my polypeptides, the separate chains, and that's what makes a functional protein. And just as an example, hemoglobin is a protein that is made up of 4 different subunits. It has 2 beta subunits and 2 alpha subunits, and that's the hemoglobin's overall shape. Its super 3D structure is its quaternary structure. So that's really all you need to know about the protein structure. Now what we need to talk about is what happens when I denature a protein. So when I talk about denaturation, that's going to be when I change the shape of a protein. So that's something that we're going to talk about in class is like melting the protein or melting the protein's face, but it's not always has to do with melting. But denaturation is anything that causes the bonds holding the 3D structure together to break. And those bonds can be your hydrogen bonds, the ionic bonds, the disulfide bonds, but anything that causes those bonds between the 3D structure, so anything in that that secondary or tertiary structure, anything that breaks those bonds is going to be denaturation. And obviously, the easiest example of this is temperature. If I heat a protein up high enough, it's going to go ahead and cause the bonds, the hydrogen bonds, between the different sections of your polypeptide to break. The hydrogen bonds break, and then that causes the protein to unfold some or fold up differently, and so I've denatured my protein. Also, pH will go ahead and do this, changes in pH and salinity, because if I look at changes in pH or changes in salinity, I can greatly affect those interactions between my positive and negatively charged side groups, those positively and negatively charged R groups on my amino acids. And so this changes the secondary structure, which remember that's the alpha helices and beta pleated sheets, and the tertiary structure, the interactions between the R groups. It changes that structure, and therefore it destroys the function of the original protein. That original protein can no longer do its job. And that's all it means when we talk about denaturation. So I hope that's helpful and that you understand a little bit about the 4 levels of protein structure. If you have any questions, just come on and see me during class or during block lunch or send me an email.